Okay. How's this? Yes? Okay, great. So I was saying that Alex hopefully will see the PowerPoint one way or the other, either through eCollege or he's had some trouble with the login. Or Monica, I think you're sending him the, thank you, the paper. So we're going to get started and then Alex just join us on the page we are. So I'm not going to share the screen with you for this, but go ahead and open your PowerPoint that you downloaded from eCollege for today. And we're going to go through that. So before that, tell me, so everyone has read the Beetle paper, correct? Yes? Yes. Okay. Good. So what I decided, so first off, you can tell me if you have any questions about that paper. So whether they're technical, specific, or overall, you can write your question in the chat, and then um, I'll answer them. So let's do just a couple of minutes for any questions from the Beetle paper. So you can write them now. Cristobal, okay, so you are not in eCollege, meaning, okay, so you were able to log into eCollege, but you did not find the PowerPoint or the Beetle paper. What exactly? Oh, okay, right. Okay, so you had it before. Okay, great. So if anybody has questions about the Beetle paper, um, you can write them down right now and I'll answer them. quickly. Okay, no questions, it seems, right? So if no, just reply no. Um, figure 6 in table 1. Okay, all right. Um, let's see. Figure 6 and table 1. 6. Yes, okay, got it. So let me see. You guys are okay, good. So those questions I will answer. Right. Okay. So I'm going to answer both Ye and Jiang your questions in through the PowerPoint. So right now, everyone's going to go to the PowerPoint. We're going to start on slide 1, and I will talk about it. Okay. Um, that's going to be the last thing we do for today. So let's see. Everybody has it open. Everybody's ready to go. Yes? Can you reply, please? Right, Shijuan, got it. So I will get to that question. Okay, so we are starting. Hopefully, Alex, you will be with us at some point. All right, so this PowerPoint is 6 years worth of climate change and ecology field research that I completed at Stanford for my PhD. And so it's a wonderful way for you guys to see a different aspect to climate change that is very much on the ground level, and I think is a really rare opportunity for you guys even before you've reached college to see the way um, science is conducted, way experiments are done, and the way a PhD is completed. And a PhD really, as a degree, is the highest degree one can get academically. But behind that, you must be able to work on a question scientifically that and specifically examine it the way that it's never been examined before. And you work for years on it. You design your own experimental procedure, and you have to find funding. You have to find a way to make it happen. You have to have collaborators. It's a very big project. In a way, it teaches you how to run your own company because you're involved from everything, from the theory to the models to the experimental work on the ground to writing the paper and getting published. And so for those of you that are interested in science further on, this is a really good example. But for all of you, this is really rare for anyone to see this kind of work right now where you guys are. And just so you know, I have to say that this material is copyrighted, so please do not share it, and please do not reproduce it. Okay, so let's start. So on page 1, this is my field work, and the image that you see is actually a boardwalk going into a natural grassland here at Stanford in California, in Northern California. And so I worked with the Carnegie Institution of Science that has one of its affiliations here at Stanford and through the Department of Biology. And so the talk, my work, you can say my life's work has been on chemical communication of invasive weeds and opportunities for control in the future. What will happen when climate change gets more and more of an issue? So if we go on to the second slide. So of course for this I'll just be presenting highlights of the work, and it's framed very much for you as a development and evolution of the research questions. Then it's really keeping kind of a detail and yet bird's eye view of the research. And it's going to give you a really good taste of the kind of research that is being done right now across the world. So going on to the next slide, slide 3. So anthropogenic climate change is affecting life on Earth. We've just seen that midway through the last century, the effect on ecosystems, natural processes, and feedbacks, plant and animal life have been unprecedented. If you go to slide 4, you see that greenhouse gases, their emissions are the highest that they have been in recorded history. Ecosystem response to climate change driven abiotic stressors. Abiotic stressors mean non-life, abiotic versus biotic. So biotic is life. So let's say herbivory, or when deer eat a plant, that's a life or biotic stressor. Abiotic stressor is rainfall, nutrients, CO2, gases. Those are they're natural, but they're not alive, so to speak. So they're abiotic stressors. They're shifting environmental dynamics, such as community composition and invasive species abundance across the world. If we go on to slide 5, invasive species, which are non-native plants, and specifically that I will be talking about, they threaten ecosystem biodiversity and stability regionally, wherever you look, globally. They successfully exclude, displace, and hybridize with natives. What you're looking at, the school bus swallowed by ivy, it looks like. It's actually a vine. It's an invasive vine called kudzu, and it's known as the vine that ate the South. So this is what the southern part of America, in certain locations, looks like. So there's evidence that climate change, of course, will facilitate invasive success. Invasives have demonstrated inherent adaptability to extreme weather events seen under climate change, and warmer winters that we're seeing are actually expanding their northern range. We go to slide 6. One of the most prolific invaders of the 21st century is a winter annual by the name of Centaurea solstitialis. It's beautiful. It's commonly known as yellow star thistle. It's native to southern Europe, and it was actually introduced as an alfalfa seed contaminant indirectly from to North America from Chile in the 1800s, it established dense local populations in California, and from then on, it invaded grasslands and rangelands used for, and then further spread by grazing cattle. Um, this weed greatly depletes groundwater. It reduces rangeland yield and quality. It's actually toxic to horses. It can actually kill horses because it's a neurotoxin. If they eat too much of it, it's over. And it displaces native plants and reduces wildlife habitat. Um, as you can see, there's a crown of thorns, and it's not very pleasant to come across. Let's just put it that way in that adult stage. So moving on to slide 7. So what you see is that today California is the most heavily infested state. Nearly all counties are colonized with over 15 million acres. Next slide. Um, you can see the map of America. So, road development and contaminated hay have led to its expansion into other states. So, it's moving eastward and northward. So, as far as New York to the east and the Canadian provinces in the north, with the largest adjoining infestation occurring right here on the west coast. Next slide. So, the sharp spread of yellow star thistle in North America, so that top graph you already saw, coincides with the recent rapid increase of CO2 levels. And from what we just learned, in a 40-year time period, roughly between 1960 and 2000, atmospheric CO2 has increased by approximately 50 parts per million, during which yellow star thistle, which we're calling YST now, its expansion grew exponentially from 1 to 14 million acres. Next slide, slide 10. So, in fact, actually, there was a global change study done right here at Stanford by Jeff Dukes et al., which from the graph you can see the invasive has been shown to grow in gray as much as 6 times larger under elevated CO2 levels projected for the future in comparison to ambient, which means natural levels. While the growth response to CO2 far exceeded, I mean, it's incredible, far exceeded those of grassland resident species who are there in that grassland. Usually you can see that in B when comparing the percent response to CO2, shown in black with that of other residents shown in gray. And so the more we know about the biology of this invasive, the more powerful its management can be. And so, to this end, that is why I chose to examine yellow star thistle, and specifically in the context of plant insect interactions as well. 11. So, insects and plants are inextricably linked. One cannot exist, right, without the other. And we're all very familiar with their interaction in the form of pollination, um, in the form of insects laying eggs on plants, And those of us who garden are especially familiar with insects eating plants. Slide 12. Um, Herbivory it's an inherent part of plant-insect interactions, and it's a very strong force for controlling plant community composition. Um, Given its enormous impact on plant livelihood and survival, plants have actually developed an incredible variety of defenses in 300 million years that we know of their coevolutionary relationship. Slide 13 is what we're on now. Plant defenses against herbivores are characterized by two major strategies: direct and indirect defense, each of which can be constitutively, meaning consistently, all the time expressed, or induced following damage of the plant. So the production of anti-herbivore traits or direct defense and signaling to the third trophic level, so meaning signaling all the way up via trophic cascade in indirect defense, can occur by physical or chemical means. So, and also all plants use chemical defenses to protect themselves from attack by herbivores and pathogens. Um, Scientists found that most chemical defenses are deployed inducibly, and they're actually, as of now, documented in over 110 plant herbivore associations. So, inducible defenses are often more efficient because when the plant is not under attack, it may reallocate those resources to growth and reproduction. So, let's take a closer look. So constitutive direct defense immediately exerts, if you look on slide 14, a negative impact on herbivores via physical barriers. So you can have spines, you can have wax, or chemical barriers such as toxic compounds produced by the dangerously beautiful foxgloves on the right there. Now slide 15. Induced direct defense actually acts upon damage, right? So examples of this include landmark work by a researcher named Ian Baldwin on inducibility of nicotine in production as an herbivore deterrent in tobacco plants. And so this is so widespread that defenses even operate underwater in marine plants. Certain seaweeds produce them as feeding deterrents on tropical reefs where herbivory is intense. Slide 16. An example of constitutive indirect defense is the symbiotic relationship between the acacia ants and the acacia tree. So ants live on and feed from the plant. And they eat these sugar bodies that the plant produces. You can see that in the photo. And they live in these hollow tubes that are part of the stem of the acacia. And in return, ants become bodyguards. So they protect their host against herbivores and competing plants. 17. So finally, induced indirect defense is where the plant, upon herbivore damage, emits volatile compounds that attract the herbivore's enemy, such as a predator or parasitoid, to defend it, actively reducing the number of feeding herbivores. So what you can see here is that volatile compounds that are They're chemical compounds that are emitted from maize when it is damaged by caterpillars, attract the parasitic wasp, which will actually lay eggs inside the caterpillar and kill it. So slide 18. So then we have to ask, how exactly do insects, such as predators, indirect defense, find their host? So what they do is they use their senses, arguably the most fascinating of which is olfaction or smell. 19. So chemical communication lies at the core of this process. It is an integral part of plant-insect interactions, and it is possible through the use of plant volatile organic compounds, which we're going to call for short VOCs. They have a high vapor pressure under normal ambient temperature, and these plant volatile odors that are smelled or read, so to speak, by insects as behavioral cues. And their perception is mediated by olfactory receptor neurons, which are located on the insect antenna. That is really what the antenna is for, just so you know. And they convert the signal, the smell that they receive, from chemical to electrical, and then it's integrated by the central nervous system. Now, slide 20. So, terpenes are the best characterized and largest class of plant volatile compounds. Apart from their pharmacological properties, plants often utilize these compounds to deter herbivores and attract predators. So we've seen them. We've seen them talked about already in seaweed. You see these compounds, terpenes, underwater. And specific terpene compounds, now moving on through slide 21 to slide 22, specific terpene compounds such as limonene, which you know, all of you know, if you've smelled lemons or any citrus, it has a very strong antimicrobial and herbivore repellent property. And then while beta-caryophyllene, which you see in the middle there, if you go to slide 23, is actually implicated in indirect defense, which attracts the parasitic wasp to caterpillar damaged maize. So this is incredible. If you think about it, all of the smells that you smell in nature that are a part of your deepest memories, right, that evoke so much emotion from within, actually serve incredible purpose in billions of fundamental interactions between insects and plants. They're a code that are read by insects, that are read by other plants too. So plants respond and insects respond to very many things. And it has even been shown as an aside, which is really interesting, that plants respond to the energy that surrounds a person. If they're growing, let's say, in a garden, if there's a lot of negative energy such as anger, fear, plants actually will wither and die. And if there is positive energy in their environment, then they flourish, which is not that hard to believe, given that that's exactly how, in a way, how it happens with us. So they're far more intelligent than we have the time to think about them. So if we look on slide 24, if ever you look closely at a plant leaf, you'll see that many terpenes, for example, are stored in and emitted from glandular hairs or trichomes that situate themselves above the leaf surface. And shown here are tomato leaves on slide 24. And they're trichomes where you can see the metabolically active storage compartment located at the tip of their hair. So slide 25. Given that herbivory and defense are dominant evolutionary forces in shaping community composition by affecting plant fitness, I wanted to understand their role in yellow star thistle invasive success up until today and their effect on YST expansion in the future. So if you move on to slide 27. So for this slide, it's the white slide with yellow star thistle in a little picture box at the top center. So to do so, I set out to answer those following questions that you see. Does yellow star thistle produce volatiles upon damage? What specific compounds are produced? And are they relevant to defense? Does the wounded plant attract predators through such compounds? And will their profile change with climate in the future? So next slide. So as we can see, and actually many of us know firsthand who have ever been in areas where yellow star thistle grows, and some of you will recognize it. Yellow star thistle is actually the poster child for direct defense with its flower bud stage. And so this is the stage that is actually most publicized and evokes mixed feelings from those that encounter it. However, yellow star thistle doesn't always look like this. Next slide. So this is young yellow star thistle, actually prior to that flower bud stage. And so the question is, what if it could be controlled at this stage by insect herbivores? It turns out that it can. So in the next slide, you see Duroceras reticulatum, which is the gray field slug. It's an invasive gastropod from Europe and a dominant herbivore of yellow star thistle, but prior to flowerhead formation. And so in fact, there was a study done here that showed that this gastropod actually accounts for over half of plant quantitative changes in grassland, which serves as yellow star thistle habitat. And so it's a major driver of plant community composition. Next slide. So if we look to the optimal defense theory, which states that plant defenses are beneficial, but they have a cost. We expect them to occur when the plant is most vulnerable, right? So such as during the early growth, when young leaves are most palatable to D. reticulatum, actually. Next slide. So the aim of my first study that I performed was to understand whether young yellow star thistle utilizes indirect defense. Next slide, and specifically whether yellow star thistle signals to predators of D. reticulatum upon damage. And so the most common natural enemies of D. reticulatum are actually ground or carabid beetles. That's used interchangeably. They actually strike at the head of the slug to immobilize it, to prevent mucus secretion, because once a slug is stressed or threatened, it produces copious amounts of mucus, so it's really hard to grasp. And you'd be surprised how fast slugs travel. If you've ever seen them, they can travel incredibly fast, and so they will just disappear. Um, So beetles, they actually strike at the head to immobilize the slug. They prevent mucus secretion this way. And so to test for indirect defense, I chose two beetle predators. Scaphanotus interruptus, which is on the next slide. It's a carabid beetle that feeds on snails. Sorry, those two slides were, so let me go back. So no, that's right. So Scaphanotus interruptus is a carabid, is one of the two. The other one on the next slide is Pteristichus melanarius. So Pteristichus melanarius is known as the dominant predator of D. reticulatum and actually a big player in slug management. You can see for both of the beetles that we've seen, those mandibles are really pronounced, right? So mandibles are they're calcified structures that act um, as as teeth or like pincers that crush, and so they're really pronounced in both. So to the next slide, you see uh, all the four players, A, B, C, and D. So we collected these players, my team and I, within this tritrophic system, and tritrophic means three levels, so plant, herbivore, and predator. Adult beetles, slugs, and yellow star thistle seeds from Stanford's Jasper Ridge Biological Preserve. It's a really beautiful preserve. It's located adjacent to the university. It's natural grassland and forest, where experiments can take place. And so we grew the seeds under greenhouse conditions and housed the other three species in temperature controlled chambers. And so this is where the beetle paper comes in. So the beetle paper is one of the first studies that I published from my research here at Stanford. And this PowerPoint, it gives you a much more clear and straightforward and simple, non-scientific form insight into this work. So, so in the next slide, so you see this gray lab space, and we actually used a Y-tube olfactometer, which you can see it's stationed right above the white paper. It's a glass Y tube It's hollow. Um, and we used it to examine the behavioral responses of two species of ground beetle to olfactory stimuli, which are emitted uh, by damaged yellow star thistle, damaged by that slug. And so what you see, if you look closer in this picture, you see a Teflon sealed jar. Teflon is an inert material because plastics also release chemicals, sadly. And in this case, it interferes with experimental work. So all plastic, Teflon is a different type of plastic that's most stable and doesn't release chemicals. So you have a Teflon sealed jar, which holds a slug damaged yellow star thistle plant. Um, both plants, so there's one in each jar, two jars. The plants have been damaged by slugs. Slugs have been removed. Plants are in the jar. So one jar is a control, so it has no damage, and the other jar has been damaged with the slugs removed. And air was released into each plant jar with yellow star thistle and then into the glass Y tube. And given the early morning activity, actually, of the carabids and the gastropod, the experiment occurred under under red light conditions. Next slide. That is what you see. So the beetle was given a maximum of 15 minutes to make a decision, and a choice was recorded when the beetle reached the end of an arm and remained there for at least 30 seconds. And so, if no decision was made within that time, it was deemed as no choice. Next slide. So, this is the first figure in that beetle paper. So how to read the bar plot. So the y-axis shows the percentage of damaged or undamaged choices that were recorded. The percentage of damaged choices is in black, while the percentage of undamaged choices is in gray. So what we found was that 60.6% of the trials for S. interruptus chose the damaged yellow star thistle odor, ultimately not showing a significant preference for slug damaged plants. It was not deemed significant enough. P. melanarius, on the other hand, which you can see on the right, made a choice for damaged yellow star thistle odor 89.7% of the time, which demonstrates a significant preference for the odor of damaged YST by slugs relative to the odor of intact plants. Next slide. So, interestingly, what we also know is that S. interruptus, who didn't show significant preference, is native to North America, occurring almost exclusively west of the Rockies, while P. melanarius, who did show a significant preference for slug damaged YST, was accidentally introduced from Europe similarly to both yellow star thistle and D. reticulatum. So, if you do one more click, you'll see that this suggests that potential indirect defense system actually may have had time to co-evolve with those non-native species that are from the same region. So that beetle that showed a significant preference, that slug, and yellow star thistle are all from the same region. And so this is significant. This is important to state that they probably co-evolved this defense mechanism. They had time to do that, and they're excellent. They're an excellent indirect defense system here where they've all come. So if we look further, in the yellow next slide, highlighting study 2. So then I asked, so if this tritrophic system shows evidence of indirect defense via behavioral assay, I wanted to know what the odor that attracts P. malarius is comprised of. So for study 2, my aim was to identify the volatile compounds emitted from yellow star thistle upon D. reticulatum damage. Next slide. So to do so, I performed a volatile assay of slug damaged yellow star thistle using a method is called solid phase microextraction. So in short, it will be called SPME. It's a device with a thin retractable fiber which traps chemicals that can be desorbed, meaning evaporated back off, on a gas chromatograph. So in this experiment, volatiles from slug damaged yellow YST leaves were collected in situ, meaning on the intact plant, by enclosing leaves with a custom Teflon bag with outlet ports through which the SPME fiber was inserted and allowed to collect the volatiles. So on the next slide, you see the GCMS machine. It's a big machine. Collected volatiles are directly injected. You can see the injector there into the machine, and they're analyzed with the help of a mass selective detector. So the way this machine works is gases go in and they move through this cyclical column, which is like a hose, tightened into loops. And as it passes through the column, the column has different polar and chemical properties. So if a compound is polar, if it's nonpolar, depending how big each molecule is, it gets separated on the column. And then it will move out of the column there at different speeds, right? So the larger the compound and the more, let's say, adhesive to the column based on the fact that it's polar, it will then come off the column slower. So that basically separates the particles based on their properties. And then it goes into a mass selective detector, which bombards and ionizes the particles and breaks them down into their smallest possible, in this case, fragments, and then checks their charge and weighs their mass, and then they come out. And so the time at which they come out, based on other known particles, you can identify that particle. A really, really wonderful system. It's quite old and has been used a lot in different forms. And so what comes out, and one of you guys had a question of the figure in the beetle paper, what is that? So this is what you get. This is a reading on the next page. You get a chromatogram. So this is a run. So these compounds have shot out, right, out of that machine. And on the x-axis, you have the time that they came out. And so based on the time and based on the fragment pattern of those compounds, and each compound is a peak. And each peak has a time that it came out and a fragment pattern. And that shows that is the way that you can identify those compounds. And so they were identified. And so what you see in A is that undamaged yellow star thistle did not produce any volatiles, but damaged yellow star thistle by the slug produced 4 compounds. Okay, they're labeled there. In the next slide, which was table 1 in the beetle paper, you have 4 volatile compounds. They're listed. And what's interesting is that all of these volatiles are sesquiterpenes. They're a group of terpenes that are a certain size, sesqui meaning 3 terpene building blocks. And so they're a group which actually prominently figures in plant insect signaling. And so the first 3 listed compounds are known to attract the predaceous stink bug when emitted by beetle-damaged potato plants, which shows indirect defense activity in other systems. Furthermore, that compound that you see at the top, trans-beta-farnesene, is the main constituent of the aphid alarm pheromone and has been shown to attract P. melinarius to its aphid prey, which is the predator P. melinarius in our system. And trans-beta-farnesene is also shown to be emitted by other plants to repel aphids. And finally, that last really long compound, the 159, is rarely described. And it may very well be a newly identified semiochemical, which means a plant insect signaling compound. Okay, and we're going to finish the presentation. So this last study, so the next slide in red, the last study that I did was basically its aim was to understand the effect of abiotic factors such as elevated temperature and CO2 on yellow star thistle volatiles. And so to our knowledge at the time, this was the first study on volatile response of an invasive plant to global changes in a field environment ever done. So in the next slide, you basically think, well, what are the possible outcomes? So if elevated temperature and CO2 will affect the volatile signal, the signal, of course, may alter the indirect defense or that very fine communication between plant insect, right? And so if volatile emission levels would increase, then the plant's indirect defense response against the beetle, its herbivory, would also increase, meaning a stronger call for help. Because if emission would increase, then more beetles would sense that emission from shorter distances, and so it would be very effective as a natural control to decrease the slug population. Hold on, there's another sound which is very strange. All right, so can everybody hear me now? Yes? Okay, great. All right, so let's go back. So change in volatile signal may also explain yellow star thistle growth under elevated CO2 because there may be a different way that resources are allocated to volatile production. So if you recall, the invasive grew up to 6 times larger in CO2 plots in the study by Jeff Dukes. And so in the next slide, this is the environment. That is that preserve, that is the natural grassland, and that is the infrastructure built there to simulate those conditions. And so, so basically, if you move on to the next slide, we germinated the YST seedlings. They were planted into ambient plots. And in the next slide, you see the setup. So the setup is you have ambient plots, nothing, elevated temperature, elevated CO2, and then both elevated CO2 and temperature. So different quadrants having different variables there. In the next slide, if you look closely, you see that plants were grown in the field under treatment conditions and sampled the same way by SPME after mechanical damage, which actually serves as a good proxy by producing the broadest range of emission. Now, if you go to the next slide, once again, you see the chromatogram that you get. So volatiles were sampled and analyzed the same way. The next slide shows the compounds we found. So we detected 14 compounds, which are listed on the x-axis. On the slide, you can see their relative abundance levels for each treatment. It's the height of the bar. And each compound has 4 bars, which correspond to a treatment. So for a compound, let's say germicrene D, you have 4 bars. Germicrene D in ambient conditions, under CO2 and temp, and under temp. And so the error bars are showing standard error. So nearly all compounds were terpenes, which is incredible. And it included the 4 D reticulatum-induced compounds we saw in the previous study. And so, so we used statistically a 2-way ANOVA approach to see the effect. And we did not find a significant effect for any of the compounds. We also tested interaction. There seemed to be no effect. And so, although there was no effect on the palette of chemicals, it is very possible that the volatile behavior may still be changing. But there is a low signal to noise ratio in the field environment. I mean, if you think about it, there's so many other things going on. It makes these studies very realistic, but it makes it hard to tease out a specific effect. And so another thing that we found, if you look on the next slide, you see a correlation matrix. What it found is a significant co-occurrence or co-emission of volatile compounds across all treatments. And what that means, so you see a gradient legend. It's a correlation coefficient, meaning if one compound is emitted, so is the other that corresponds at a right angle. If the correlation is at 0.5, it's significant or higher. The p-value for that is incredibly small. So what you see circled are some of the most significant compounds correlation, so 0.75 and up. And the strongest correlation was between germicrene D and bicyclogermicrene, which were both present in high levels and, if you remember, are induced after slug damage. Now, what this means is that those compounds that are co-produced, they are most likely produced by the same terpene synthase enzyme. So that is another novel finding. The co-emission may be a mechanism by which volatiles are emitted simultaneously, which is very important for signaling predators, right? If they utilize a blend of volatiles, it means that compounds within a blend have to be detected by those olfactory receptor neurons at the same time to be considered as coming from the same plant, right? They have to be emitted simultaneously because that's the signal. That's what the insect is reading. And so overall, in the next slide, our studies found that yellow star thistle does produce volatiles upon damage. it identified the induced volatiles, and it found that wounded yellow star thistle does attract predators via compounds, which serve as indirect defense cues, and detected no direct effect of elevated temperature or CO2 on those volatiles. So if you look in the next slide, if we step back, we see that indirect defense may have played a role in yellow star thistle's invasive success up to the present. And so as far as the future, if we look at the next slide, we know that CO2 facilitates rapid growth, right, of YST. And if we look one more slide forward, we also know that VOC production and storage structures, right, the leaf trichomes, they have been shown to increase with leaf surface area. And you can see that here there's a positive correlation of terpene-containing glandular trichomes with leaf area. And so if CO2 may provide a mechanism by which faster growth will facilitate indirect defense and thus yellow star thistle's success. So in this scenario, if you go to the next slide, larger leaves may emit more volatiles upon induction, which will then produce a stronger signal to beetle predators protecting YST. So it reinforces a cycle. It reinforces a cycle where elevated CO2 increases YST growth, which in turn elevates the induced signal upon damage, and that facilitates YST expansion. And so in the final slide, if you look, the next slide, it's again the picture of America. Based on the available evidence, yellow star thistle will continue its expansion northward and eastward from California. And if you click, you'll see D. reticulatum, which is actually expected to expand or at least shift its range northward by the next century. And therefore, in regions where D. reticulatum is present, the ability of yellow star thistle to launch during early growth will depend on the effectiveness of its defense, which in the illustrated scenario may increase, right, due to a larger leaf area under elevated CO2. So that brings it to a close. That is the work that that myself and my colleagues and collaborators have been doing, and we will actually visit this site when you come. And hopefully you won't encounter rattlesnakes like those that we had. You will see that pictured towards the end of your slides. If it's there, it should be. What we saw was actually a rattlesnake. Rattlesnakes are very common here, especially in the summer. They come out to warm their bodies. And what we saw was a rattlesnake had just killed a juvenile ground squirrel, and 10 minutes later, that ground squirrel was inside the rattlesnake. So that's another reason for closed-toed shoes when you guys come. So I will see you tomorrow, regular time, and we will move on from there. If you have any quick questions, you can write it in the chat, but otherwise we are ready to go. All right, everybody else can turn off their systems. Thanks. Carolina, you can go ahead and just write the conscious diary, and I'm going to take a very thorough look this weekend, and I'll let you guys know at the beginning of the week. So if you're doing enough, don't worry about it. The format, if I'd like something different, I'll tell you beginning of next week. So don't stress about it. And Lily, so 15 minutes was chosen as a scale because that's enough time for them to make a decision. Beetles uh, can actually they take time at the entrance to the olfactometer, and they take time to sense the chemical compounds, and then they need to make a decision, and then you need time left over to make sure that they indeed did make their own decision. And so 15 minutes is an optimum simply because of the animal species that you're dealing with, the insect. Um, so it's more of a behavioral point rather than like a strict um, scientific point. We wanted to give them the most amount of time to really kind of make sure that that was their decision, that we didn't write down a decision too quickly. So those are just details that change with experiment and species. And as far as part two, we can talk about that when you come back on campus and when we meet in person. All right. Yeah. Monica, our hike will most likely be on Monday the 17th. So it's the final week, week 6, and it's going to be a Monday. So that's when things will happen. All right. We'll see you guys later. Thanks. Bye.